Good morning, everybody. It's 9 a.m. here in San Diego, so we will begin today's webinar. Thank you very much for joining us today. Today's webinar is on PROTAC modeling in ICM-Pro. This webinar is being recorded and will be uploaded onto YouTube. Before we carry on, I just want to express our support for our brave friends and colleagues in Ukraine, where there are some amazing scientists and exceptional chemists. I guess one way to support them is to carry on using their services as normal. And we're grateful that some of our Ukrainian ICM users are able to join this webinar today. Thank you. So just if you're not familiar with MolSoft, we were founded in 1994 by Ruben Abagan. Our principal scientist is Max Totrov. Eugene Rausch is our principal developer, and Paolo Lam is a senior scientist here, does a lot of the development. So we've, over the years, we've had long partnerships with big pharma like Novartis and academic labs like the Structural Genomics Consortium. And we've competed in modeling and docking challenges over the years. Most recently, we outperformed many of our competitors in the D3R docking and screening challenges. The software we provide is called ICM-Pro. It contains ligand protein modeling, such as PROTAC modeling, ligand docking, covalent docking, protein-protein docking, template docking, fragment docking, and things like that, as well as a full suite of cheminformatics tools for drug design and bioinformatics tools as well. My name is Andrew Ori, and if you have any questions after the webinar, please feel free to email me or email info@molsoft.com. As I mentioned, this webinar is being recorded, so we will upload it to YouTube, to our YouTube channel. If you just search MolSoft on YouTube, you'll find it. And we have some manuals as well. I can send these links after the webinar. If you have any questions, please type them in the chat or the Q&A section of this of the webinar, of the Zoom webinar panel. So just a brief introduction to what PROTACs are. I'm sure you know what they are, otherwise you wouldn't be here at the modeling webinar. But just to sort of highlight, the PROTACs were first described by Sakamoto and Desai and Cruz lab over 20 years ago now, in 2001. TPDs, target protein degradation, is an approach that is very popular nowadays. Substantial interest for targeting drug targets which maybe prove difficult with more traditional drug design methods. So there's targets which might have, maybe intractable because their active sites are shallow or difficult to bridge with small molecules. So they offer a new opportunity for drug design. So what are PROTACs? They are hetero-bifunctional molecules. They are two ligands joined by a linker. One linker recruits the target, and the other linker recruits an E3 ubiquitin ligase, which induces ubiquitination and then degradation by the ubiquitin proteasome system. And then the PROTAC can be recycled. A very interesting review I just found a few days ago was by Craig Cruz's lab, where they talk about the last 20 years of PROTAC discovery. Sort of focuses a bit more on the sort of the industry activity in that area, and sort of maybe the future of PROTACs. And it also describes also so in clinical trials, there are two PROTACs that are in clinical trials, ARV from Arvinas, ARV110 and ARV471, that target the androgen receptor and the estrogen receptor. These two. So these are in clinical trials at the moment. So in ICM, our modeling procedure is as follows. So you need to start with the structure, the target structure, ideally with a ligand already bound, maybe an inhibitor bound or some design. And then you have the Celebron or VHL component with their respective, so you have the respective binding moieties in each. Then the PROTAC, so these are individual PDBs. So the PROTAC, or it could be models, in fact. So the PROTAC molecule is then modeled into the first complex and then into the second complex using the ligands in the in each of the site, the target and the E3 ligase as a template. We then use internal coordinate mechanics, Monte Carlo simulation, biased probability Monte Carlo simulation. I can send links to the papers that describe that method. There are webinars on using the simulation method also on YouTube. But so basically the simulation samples the torsions and optimizes the tripartite complex. So you've got three elements: the PROTAC, the target, and Celebron or VHL. While it's optimizing and sampling, the ligand is obviously fully flexible. The target and Celebron are being modeled using our protein-protein docking methodology, and also the surrounding side chains are fully flexible. This results, the resulting ensemble, it generates a number of different conformations, and then the low energy conformations are re-evaluated using our scoring function for protein-protein docking. And I can send you the papers for that after the webinar in the follow-up email. So in this webinar, we will be focusing on one example primarily, but we can offer you some other ones if you're interested. The one we're going to do today is an interaction between the development of PROTAC between the Bromodomain and Celebron. The Bromodomain is in complex with an inhibitor JQ1, which is from the Structural Genomics Consortium. It has the PDB file 3MXF. The Celebron is ubiquitin ligase, the E3 ubiquitin ligase, and in complex with thalidomide with PDB code 4CI1. And then we will dock this PROTAC, model the PROTAC into the complex, and then we'll be able to compare it with the known complex that's published with the PDB code 6BN7. So to run the modeling, first you need to have ICM-Pro full package, which you can download from MolSoft.com/support. I think I sent some instructions. If you don't already have the software, I sent some instructions yesterday. A lot of people already got the 14-day license, but if you haven't, it's fine. We can send it to you after the webinar. In the very latest version of ICM, you'll find this modeling script in the /bin directory of the ICM distribution. So if you, in Windows, for example, in C drive, program files, MolSoft, ICM-Pro. There's a directory called bin. You find it there. If you haven't got the very latest version, I sent it by email yesterday, the script. So practically, to run the script, you call ICM, and then you call the modeling script, which we name PROTAC model.icm. And then you need to have the target protein as an ICM object file .ob, the E3 ligase as an ICM object file. So I'll show you how to do this. You read it in PDB and then convert. Then you give an output, which is, you can see that the extension is ICB, which is our MolSoft's ICM binary. So it's going to save everything in this output file. You then provide the PROTAC in STF format. And then there are some options. So there's a parameter for effort. So by default, the effort is 1. This value changes the length of the simulation. So for more extensive sampling, you may want to increase the effort to 3 or longer or more, but the longer, the higher it goes, the longer the simulation will take to run. This option SCS rad equals, and then you enter a real value. We suggest this is basically a radius around which the side chains will be fully flexible. So we suggest a radius of 8.0, 8 angstroms. And then if you're in a situation where you're comparing your where you're comparing your model with a known complex of the tripartite complex, so if you have you can just provide a reference structure to compare your model with one from the PDB. You use the option ref structure equals, and then you provide the PDB code, the chain that you want to compare, and the ligand, the PROTAC that you want to compare. You don't need to read this PDB into ICM. When you have this option, it will read it automatically for you, providing that the PDB code is correct and the chain IDs are correct. So in our example, you would have this should be one single line. You would call ICM, call the script. Here we have the Celebron 4CI1 object. Here we have the Bromodomain 3MXF object. Here we have our output file, which can be called anything you want. PROTAC equals this ligand in SD format. We are comparing our results to the complex of Celebron and the Bromodomain, which has PDB code 6BN7, the B chain and the PROTAC. We're using the suggested radius for the side chains, and we're just going to output when the simulation runs, it has many lines of output, and just for reference, we're going to keep it in an output file, but the actual 3D complexes will be saved in this .icb file. And from this ICB file, you can save as PDB and read into other software if you need to do that. Okay, so we're just going to go through the steps of how to do this. So the first step is to read in the target, load the target structure and the Celebron structure as well. So in each of these slides, I show you how to do it in the GUI as well as on the command line. So to read in a PDB file, you go to the search button here, PDB search, And there's a drop-down menu. Using that drop-down menu, you can search other databases like Kemble. You can and other databases that we have linked directly to ICM. You can enter the PDB code. You can see, you can also search the PDB by chemical structure if you want, but we're just going to, in this example, use 4CI1, which is our Celebron molecule. So you can see this protein has two protein chains. One is Celebron, and there's some. Then when the ligand bound to Celebron is thalidomide. There are water molecules there, obviously, and zinc molecule. We'll be removing some of these things in a bit. So if you want to do this on the command line, so to read in our Bromodomain, for example, we type read PDB and then use the quotes 3, I think it's 3mx. I just need to double check. 3mxf. And that will read in the Bromodomain. So now we have two objects in our ICM workspace. The next step is to delete molecules in the PDB that are not relevant for what we're doing. So we want to make this, it's challenging enough to do protein dogging as it is. So we want to make this as simple as possible. So we want to remove any other chains that are involved in, that are not involved in the complex. So in this example, we just want for 4CI1, we just need the Celebron and the thalidomide. So you can just delete, right click and delete molecules that you don't need. We don't need. If you use shift, you can select a contiguous list of molecules and then delete, or you can use control and select a non-contiguous selection. So you can also use the command line here to so for 3mxf we just need the Bromodomain shown here and the ligand bound. So in the command line, you could just use delete a_3mxf dots, but not the A chain or the AJQ1 ligand. The exclamation mark means not, so hopefully if I've done this right, it should delete everything. Yeah, so now we're just left with the two molecules. So there's plenty of webinars online on how to manipulate the graphics if you're not familiar with the graphics in ICM, but just make a selection and you can center using the center button here. And we can see this is the thalidomide. We can see the interactions of the thalidomide with the E3 ligase by just right clicking on the ligands, the ligand pocket interactions. So you can see the key residues, and we can make a slide of that if we're interested in going back to that. And then for the same for the Bromodomain, we can just double click here, center, and if you're interested in the key contacts between our ligand in the Bromodomain, we can just go pocket, ligand pocket interactions, save it as a slide, and then you can just toggle between those two images if you want to go back to how it looks. But that's I'm going off topic. That's a graphics side of things in ICM. So now we have the molecules. We have everything we need apart from the PROTAC so far. But first we need to convert our PDB structure into an ICM object. So when you convert to an ICM object, there are, what it does is it basically adds, so the PDB structure obviously doesn't have hydrogens, so ICM adds the hydrogens. It assigns formal charges and partial charges. And then there's some options to evaluate some the orientation of some of the side chains. So for example, for histidine, it samples the 3 protonation states of histidine and the 2 rotations of histidine. It does the 180-degree flip of asparagines and glutamines in the pocket. And there's also an option to if you ideally you're not using a very low resolution structure, using high resolution structures. So in most cases, the occupancy of the side chains atoms in the pocket are pretty good, and the crystallographer has been able to model those into the pocket. But if there are atoms missing, you can choose to either hide them, or if you uncheck this option, you can build them, and it will build the full residue side chain structure. So to practically do this in ICM, we go back to our 2 objects. We have the Celebron and we have Bromodomain. We convert by right-clicking on the name of the object and choose Convert PDB. That gives you the options. We've already deleted waters, but you can, if you're doing it for another project, you can choose to keep tight waters of 3 or 4 hydrogen bonds. Once you've added the hydrogens, you can optimize them, and then the asparagines, the glutamines, and histidines, optimizing the hydrogen bond network for these side chains. There's other options like replace the original, which is sometimes a good idea to do if you don't want too many objects in the ICM workspace. The option I just discussed, the hide missing side chains, and you can also, using our built-in pKa model, you can calculate the charges of the ligands in the ligand in the pocket. So yeah, these are the options, and you just go OK to run that. Okay. So now you can see that here it no longer says it says ICM here and X-ray here. This is still an X-ray structure. This is now an ICM object. So for both of, you'd have to do this for both of these objects. For both of these, convert to an ICM object. And then we need to save the object. So ideally, to run the script, you need to save everything. It just runs better if you save everything in the same directory. Then you don't have to give the path to the individual objects. So we'll just save this object. If you right click on the object name, choose save as, and then you can save it in the in a directory. And you can see here I've already saved. I saved the 4CI1 as an ICM object using the file type here object, and just giving it the prefix CRBN so we know what it is, and BRD4 here. So those are the two objects. So now we have 2 parts of the 2 of the 3 things we need. The third part we need is the PROTAC itself. So that you're going to model into the complex. So you can just, if you already have the, actually, sorry, before that, with the conversion, if you already have your structure converted in another program, for example, and you don't want any changes, you can just uncheck everything and it will just keep it the same as it was. Likewise, if you have a chemical from ChemDraw or something, you can just save that in SD format in the directory where you saved your object files. But if you want to use ICM to do that, I'll just quickly show some ways. One way is to read in, if you have the PROTAC, this is the PROTAC complex, full complex in the PDB 6BN7, and it has the PROTAC bound here. So one way to extract from the PDB this ligand is to right click on the ligand and choose the option to extract ligand here. And you can keep 3D coordinates or 2D coordinates, and it will make it into a table here. And then this chemical table can be saved as, if you right click on the header of the table, you can save as SD file. You can go back to the setup before and save the SD file in that directory. Okay, the other way would be to sketch. You can sketch, use the sketch button here and sketch your PROTAC and save it as an SD file. Or you can read it as a smiles, or however way you have it. If you have it in smiles, you can just go edit and add smiles and add the smiles string here, and it will save it. Okay. So basically now we have all the components we need to run the script. So we need to save the PROTAC model.icm script to a directory or give the path to where it's located. We have our object of Celebron for CI1. We have the object in ICM object of Bromodomain. This is an output file, so it can be called anything. We have the PROTAC in SD format, and then we have our options. I'm using a Mac, so I'm using the Unix terminal on the Mac. But if you're using Windows, it's slightly different because you need to run the Windows terminal. So you would go to Start menu, I think, and then just enter command.exe, and that will open the terminal. And then you can do exactly the same syntax, except to call ICM, you need to call the ICM console 64.exe, and then the arguments after that. So as I mentioned earlier, we're using a Monte Carlo bias probability Monte Carlo simulation. Generally, with these kind of simulations, it's best to run multiple times. So and so we would recommend to run this script at least 5 times. And then to do that, you would just need to change the suffix here. So if it's the second one, just change the suffix of the output file to 2, for example, would be easiest, the ICB file, and also change the suffix of the output, and then just rerun the script. So I'm just going to show you how to run it, and it's basically just pasting the commands, but then we can go there. So here I have hopefully this is shown okay on webinar, but on the Zoom, but we have all the components that we need in this one directory to run the script. Here I have the script, just copying it. So go back to this directory. So we just copy that, copy this, and press enter basically, and then it'll start running. So if we go to the, so if we go back to this directory, PROTAC demo, I think it's here. Yeah, we should now start seeing the output file that we have start filling up with output. sorry. So this is the output file. So you could, this is just what's generated using in the simulation, or the simulation details, which you can, I can send more information, but if you want to read it in more detail. But really the key for in terms of the 3D structures, the ICB file, the output ICB file is the key one. Okay, so when you finish running it, you will then have, if you run it 5 times, for example, you will have 5 ICB files with different docking complexes in each one. So you want to basically combine them into one single file. So you would have, so in our example, we called the ICB files Celebron, Bromodomain, and then 1, 2, 3, 4, 5. So we can use an asterisk wildcard to process them using this macro process PROTACS sims. And that is run inside ICM. So the wildcard will process all your output files into one single file. So if we go to the, I think this is the results directory. Yeah. So here I ran that. So I have 5 output files in ICB format, 5 ICB output files. And so then if you run ICM, then, or in the GUI, you can use the option process PROTAC sims. This is in the ICM shell, not in the Unix shell, and then give the name star, and then that will process them. It takes a few minutes to run to process. So I won't show you, but so when it's processed, you'll get all of your models in one single ICB file, and it will be called PROTAC model all.icb. When you open that PROTAC model all.icb file, you will then see the results table and a plot if you've compared it with a reference structure. So the results table is ranked by the energy, which is the total energy of the complex, and this really is the main scoring of the complex. So the lower the energy, the better. It will report the RMSD, and there's a column to add comments. There's an energy of force field, there's a surface energy term, there's a van der Waals interaction energy term, electrostatics, and the energy of the side chains. Let's look at that in ICM. Going back. So if you open once you've processed them into one file, PROTAC model all.icb, you will see something like this. So this is our table, as we talked about, and you can load. So this is ranked by energy. So you can load a complex by double clicking, and it will load it into the ICM workspace. So it's called conf3 because it's third in the ranking here, and this is one's called conf2 because it's second. And so you can see as you load them, you can see the differences of your complex as it's loading. So you can sort by energy, you can sort by RMSD, as well, or any other column here by energy. And if you have a reference structure, it will build this plot automatically, which is fully interactive. So here we have the low RMSD and low energy conformations. You can select those. You can see it's selected here, so you can maybe extract those, or you can just double click to load them. If you've loaded them, you can save as a PDB by right-clicking on one of the models and choosing Save As PDB. Choose the option Save As here, PDB. Then there's some other options you can use that are inbuilt into ICM just to look at the. I was going to delete these because there's so many of them. Just to look at the interaction of the PROTAC with the ligand. So it's going to load this one. So you can, for example, you could just right click on the PROTAC and choose pocket, ligand pocket interactions. That shows you the key interactions of that PROTAC molecule. You can display hydrogen bonds by going to the display tab here and clicking on the hydrogen bond atom. And next to that is the pi-pi interactions. If they're pi-pi, you'll see those actual interactions displayed. You can also analyze the contacts of the Celebron with the target protein, or between the ligand and the target, and between the PROTAC and the two targets. So one way to do that would be to select the PROTAC, for example, and then go to Tools, Tools Analysis, and then choose the option Contact Areas, and this will just go OK. So this will give you a table with the contact area of each residue, the exposed area, the percent of the side chain that's buried, and the distance to the selection. It also gives a scale to the side chain representations for how much. So this side chain makes the biggest. This tryptophan makes the biggest contact with the ligand, so it's in thicker stick. So the thicker the stick representation, the better. You can also view the pocket receptor pocket for this, so you can see the where the coloring is by the pocket. So this represents hydrogen bond acceptor, and blue represents hydrogen bond donor. Green is hydrophobic, and white is lipophilic. And if you want to, you can display that here. You can also display maybe the ligand surface. Might take a little bit longer to calculate. Yep, it does. Yeah, so that's a different. That's the opposite representation, and it gives you a better representation of where there's cavity to build into, or make modifications to your ligand. You can use the ligand editor to do that. There's other webinars online on our YouTube channel on how to make modifications to ligands, how to dock them, how to screen for substituents, and things like that, and how to also link fragments as well. There's different webinars. Yeah, so those are the key things. So you have it's the results table, and the key column is energy. So if you want to compare one of the better RMSDs, we can load in the or any, actually most of these are pretty decent top hits, but you can load in go to the search and load in 6BN7, which is the crystal structure of this complex. So I'm going to undisplay that surface. So this is the crystal structure 6BN7, which we loaded earlier to extract the SD file from. But if we superimpose this on one of the docked conformations, we can just see them superimposed. To superimpose, you make a selection, use the control button or command button to make a non-contiguous selection. So we've made a selection here, made a selection here by clicking, and you can see it's highlighted blue, so it's selected. Then you can go to Display tab and use the option to superimpose. And it's best to keep the docked complexes as the static object and superimpose the crystal structure onto that, just so things don't get out of. So yes, so here we can see the. So red is the crystal structure and green is the docked. Sorry. The same with the Bromodomain. So Bromodomain's here. So this is the blue red. So this is a comparison of the crystal structure with the docked molecule. You can zoom into the ligand as well and see the comparison. You can probably change the way it's superimposed because it's superimposed. You just make a selection, then it will superimpose whatever you have selected. So just go OK, and that will superimpose the pocket region, which looks a little bit better. It's just shown here. Okay, so thank you very much for your attention, and thanks very much for your interest in the webinars. And if you have any